Good morning, afternoon, probably. So my name is Silvia Restrepo. I'm the new president of BTI, and I'm so blessed to be here as the president. So it's good I come at the end, because I come from a country that has a lot of cassava, physalis, and a lot of parasitic worms. So I can talk about those. But today I'm going to talk about my research. I'm not going to go into much details because I have to divide my time between my research and some, and I want to tell you what we are currently doing in BTI and like how the near future looks like. So I'm a plant pathologist. Actually, I was here in Cornell a long time ago, 20 years ago. I was a postdoc in William Fry's lab, and we studied the diseases of potatoes, Phytophthora infestans in potato. I continued that research in Colombia. So basically, I studied the crop losses due to diseases. I work with potatoes, tomatoes, and a lot of different solanaceous crops, including physalis. I chose the physalis way of saying physalis, or uchuva, as we call it in Colombia. And my idea is if we understand the enemy, if we understand the pathogen and its diversity, we can better fight it back. So what I do is I use a lot of molecular markers. I extract a lot of stuff or DNA from the plants, as Lucas said, and we analyze the diversity and focusing on the biology of the pathogen. So I focus more in the pathogen side than in the plant side, but I work with breeders trying to develop resistance to the different diseases. So I study the genetics of the pathogen and work with breeders. So what I do is I love to go to the field. I'm an outdoors person, so I go to the field, and this is a field in Colombia. It's so challenging because here you can see, where is that? Here. So here is the potato field, but here in the grower's garden, you can see a lot of different reservoirs of the pathogen. So it's challenging, and actually you can see physalis right here. I lost my, here. So physalis is one of the hosts of Phytophthora infestans. It's right there, and also with tree tomato and some other solanaceous crops that can be the reservoir for the pathogen and for the growth. So I go to the field, I collect a lot of samples, I go back to the lab, extract their stuff, but also study the host of the pathogen. So for only this pathogen, Phytophthora infestans, we found in Colombia a lot of different hosts that were not reported before. Of course, potato and yellow potato that we call Solanum fureja, tomato tree, tomato lulo, that is Solanum quitoense, uchuva, that is, I call it Cape gooseberry, but it can be goldenberry, Physalis peruviana, and some wild species like Solanum viarum here in what we found by the road. So I studied the diversity, the genetic diversity of the pathogen. So here you see the map of Colombia, and what do you see in different colors is the different variants, the genetic variants of the pathogen. Some of regions of Colombia have a lot of diversity of the pathogen. Actually, we found out that in this region we had 2 species of the pathogen, no one but 2 species, close to Ecuador and Peru. So we study the diversity, and then we go back to the field and study and do some assays in the field to look for sources of resistance to these different genetic variants of the pathogen. Actually, this is in Freeville here in New York, where we conducted some research with Bill Fry and Chris Smart. And then we selected some varieties. I work with breeders, mainly with Carlos Nustes in Colombia, and we just recently deployed some resistant potato varieties in Colombia. And of course, I do that with my team. This was probably one of the last pictures I have of my team in Colombia. And my new postdoc, the only member of my lab here in BTI, Juliana, is actually right there. She was doing her master's in my lab back in Colombia, like 5 years ago or something like that. Okay, so basically, I would like to imagine a future where we don't have these threats from the diseases. And we also aim not only to reduce the crop losses, but also to reduce the use of inputs and dangerous fungicides and other pesticides in our crops. But let me switch now to BTI. So my other hat. I have the researcher hat and I have the presidency hat. So, and today we have reflected on BTI's remarkable century. And we have reflected in our legacy that is so incredible. And we are very grateful to our alumni. But in this moment, I wish to acknowledge all these contributions from our alumni, but in particular, Alan Renwick, who passed away 2 days ago. Alan participated, and this poll told me that, participated in 65% of BTI's history. And he made significant contributions, George mentioned some of them, to BTI's history, and especially to pest control and environmental challenges. So I would like to ask you, and please join me in a moment of silence in Alan's memory. Thank you very much. Alan will be remembered here in BTI. Okay, so we have been working in this near future adventure of BTI. Of course, we think in the next 100 years, but we will probably not be around. So we are thinking in the near future, in the next 2 or 3 years that we have ahead. So we have worked in this new strategic plan, and we have key aspirations, and we would like to work in the 3 most important pillars of BTI. The first one, and you heard today, amazing science, is the discoveries and the fundamental science we do at BTI. So we would like to foster pioneering research, develop a strong translational research program, and we have Julian here, and you talk about patents. Well, 2 persons to your left, to your right is Julian, who is in charge of all the patents in the Institute. So we would like to develop the strong translational program and, of course, expand our global partnership, especially our international partners. The second pillar is our organizational optimization. We would like to be efficient, to have streamlined processes, to have also to promote transparency throughout the Institute and a democratic structure. And the third one, of course, is education and outreach. And we are thinking in this new generation of scientists that we have here in BTI, that we have all the grad students, but during the summer we have these amazing high schoolers and undergraduates that come. This last summer we had, I don't know, Delaney's around 45, I think, 45 undergraduate students and 4 high school students. So please scan this QR and explore our strategic plan. So I will give you a minute to scan the QR so you can go after this amazing journey today and go through our aspirations, actions, activities, and indicators for the next 2 or 3 years. And if you like to contribute to any to our different actions, please write to me or write to any person here in BTI, and we can include you in our discussions. And we also have right now this campaign, what we call the First Million Endowment Campaign. So the Cambridge Foundation, from one of our members of the board of directors, pledged to match all gifts that we received during this year. He pledged to match until up to 500,000, and that's why we are calling the First Million. So any dollar you give us today will be matched by him. So it's a wonderful campaign. Right now we are at 65% of our goal, so not bad for a year at BTI. And why support this campaign, or what are you supporting? Our groundbreaking research, the translational program, and our education and outreach programs to help the new generation of scientists. So please join us with your contribution.